Hello and good morning. This is morning for me, but probably by the time you see this, it won't be. We are going to be reading from chapter 3, Cell Structure and Function. And that is right here. If you go to your book here, you'll see it here starting at page 64. We're just going to read a few pages today. And if you opted to read by listening, then you will need to take some notes. Let me turn to the page. Okay. So chapter 3:1, cell theory. And we're just going to skip around. So we'll skip the first paragraph. So main idea, early studies led to the development of the cell theory. Almost all cells are too small to see without the aid of a microscope. Although glass lenses had been used to magnify images for hundreds of years, the early lenses were not powerful enough to reveal individual cells. The invention of the compound microscope in the late 1500s was an early step toward this discovery. The Dutch eyeglass maker Zacharias Janssen, who was probably assisted by his father Hans, usually gets credit for this invention. A compound microscope contains two or more lenses. Total magnification, the product of the magnifying power of each individual lens, is generally much more powerful with the compound microscope than with a single lens. Discovery of cells. In 1665, the English scientist Robert Hooke used the three-lens compound microscope, shown in figure 1.1, to examine thin slices of cork. Cork is the tough outer bark of a species of oak tree. He observed that the cork is made of tiny, hollow compartments. The compartments reminded Hooke of small rooms found in a monastery, so he gave them the name cells. The plant cells he observed, shown in figure 1.2, were dead. Hooke was looking only at cell walls and empty space. Around the same time, Anton van Leeuwenhoek, a Dutch tradesman, was studying new methods of making lenses to examine cloth. As a result of his research, his single lens microscopes were much more powerful than Hooke's crude compound microscope. In 1674, Leeuwenhoek became one of the first people to describe living cells when he observed numerous single-celled organisms swimming in a drop of pond water. Sketches of his animalcules are pictured in figure 1.2. As people continued to improve the microscope over the next century and a half, it became sturdier, easier to use, and capable of greater magnification. This combination of factors led people to examine more organisms. They observed a wide variety of cell shapes, and they observed cells dividing. Scientists began to ask important questions. Is all living matter made of cells? And where do cells come from? Cell theory. The German scientist Matthias Schleiden also used compound microscopes to study plant tissue. In 1838, he proposed the plants are made of cells. Schleiden discussed the results of his work with another German scientist, Theodor Schwann, who was struck by the structural similarities between plant cells and animal cells he had been studying. Schwann concluded that all animals are made of cells. Shortly thereafter, in 1839, he published the first statement of the cell theory, concluding that all living things are made of cells and cell products. This theory helped lay the groundwork for all biological research that followed. However, it had to be refined over the years as additional data led to new conclusions. For example, Schwann stated in his publication that cells form spontaneously by free cell formation. As later scientists studied the process of cell division, they realized that this part of Schwann's idea was wrong. In 1855, Rudolf Virchow, another German scientist, reported that all cells come from pre-existing cells. These early contributors are shown in Figure 1.3. This accumulated research can be summarized in the cell theory, one of the first unifying concepts developed in biology. The major principles of the cell theory are the following. Number one, all organisms are made of cells. Two, all existing cells are produced by other living cells. And three, the cell is the most basic unit of life. I encourage you to examine Figure 1.3 and look at the contributors to cell theory, which I will not be reading. And the last page we're going to examine today is prokaryotic cells lack a nucleus, and most internal structures of eukaryotic cells. The variety of cell types found in living things is staggering. Your body alone is made of trillions of cells of many different shapes, sizes, and functions. They include long, thin nerve cells that transmit sensory information, as well as short, blocky skin cells that cover and protect the body. Despite this variety, the cells in your body share many characteristics with one another and with the cells that make up every other organism. In general, cells tend to be microscopic in size and have similar building blocks. They are also enclosed by a membrane that controls the movement of materials into and out of the cell. Within the membrane, a cell is filled with cytoplasm. Cytoplasm is a jelly-like substance that contains dissolved molecular building blocks such as proteins, nucleic acids, minerals, and ions. In some type of cells, the cytoplasm also contains organelles, which are structures specialized to perform distinct processes within a cell. Most organelles are surrounded by a membrane. In many cells, the largest and most visible organelle is the nucleus, which stores genetic information. As shown in Figure 1.4, cells can be separated into two broad categories based on their internal structures: prokaryotic cells and eukaryotic cells. Prokaryotic cells do not have a nucleus or other membrane-bound organelles. Instead, the cell's DNA is suspended in the cytoplasm. Most prokaryotics are microscopic single-celled organisms. And eukaryotic cells have a nucleus and other membrane-bound organelles. The nucleus, the largest organelle, includes the genetic information. Eukaryotes may be multicellular or single-celled organisms. Okay. And then 3.1 formative assessment, which I will skip. And that's the end of 3.1. I'll load 3.2 tomorrow.